immunology. Right now, we just started immunology chapter and we started reading about it. Right. In the yesterday class, friends, we have explained about all the terms: immune, immunity, immunology, and we have also explained about immune system and immune response. And I hope every student remember, right? Effector response and memory response, not to get confused. Later, I have explained to all of you the differences between the innate immune system, adaptive immune system. Every student very quickly answer my question. Among the innate and adaptive immune system, in which immune system antibodies are not produced? Everyone type the answer, mute your mics. Every one of you answer Sujata. In which immune system the antibodies are not produced? Very good. The antibodies are not produced in the innate immune system, not to get confused with that particular point. Perfect, perfect. So later we started understanding what, friends, the different cells of the immune system. If you quickly recall what we discussed, friends, bone marrow is the area where the immune cells will be produced. Bone marrow also called as what cell, friends? Hematopoietic stem cell. What is heme? Heme means blood, poietic means production. So wherever the blood cells are producing, friends, that is called hematopoietic stem cell. What I have explained to all of you, hematopoietic stem cell divided into lymphoid progenitor, myeloid progenitor, erythroid progenitor. Every student answer my question. Give me the name of the immune cell which is synthesized from unknown precursor. Radhika, Srinivas, give me the name of the immune cell which is derived from the unknown precursor. No, Venu. Everyone, forgot everyone. Yesterday only, no, we discussed. Very good. It is a mast cell. Mast cell is the one which do not have what, friends? A specific kind of a precursor molecule. So it is actually derived from the unknown precursor only. So we just started discussing about the lymphoid progenitor. Now you people has to answer me. In the yesterday class, I have told to all of you see, friends, any chapter I will teach very fast, but not immunology, because immunology is a very depth concept. I will teach slowly, but I will teach extremely effectively. By that, you never forget. Now listen, B lymphocyte, T lymphocyte. If every student quickly recall, B lymphocytes matured synthesis and maturation in the bone marrow. Whereas, friends, what about the T lymphocytes? T lymphocyte synthesis also, maturation also. See, synthesis occurred in the bone marrow, but where the maturation occurred, friends, in the thymus. In the next classes, we will study why it is getting matured in the thymus, because it undergo the selection process. Every student answer my question. Among the B and T lymphocyte, which is the most common surface marker? What is the most common surface marker that is involved in the signal transduction process? We have studied. What is that, friends? CD47, not to get confused with that particular point. And friends, later we all people started studying what, friends? We all people started studying different type of surface marker. Not to get confused, friends. B cell contain B7 surface marker. That B7 surface marker associated with surface marker CD28 surface marker, which is present on the T cell. Not to get confused with this particular point. Okay, now all the students listen to me. In the yesterday class, we just stopped while studying the T cells. Okay, now every one of you please pay attention. Try to understand it very correctly. T lymphocytes synthesized in the bone marrow, matured in the thymus. Undoubtedly, friends, how did you categorize the T cells also? Undoubtedly, friends, T cells also you categorized based on the surface marker, based on the function performed by them into T helper cell, T cytotoxic cell, T suppressor cell. So we categorize it into three types based on the surface marker or the receptors, as well as based on the function performed by it. What we discussed, very important statement. This is the most important one. which T cell, they can ask the question in the exam, which of the following T cell is compulsory required for the production of antibodies to the B cell? Every B lymphocyte compulsory require T cells. Especially which T cell? T helper cell. See friends, you can ask me, sir, why only T helper cell? Because friends, T helper cell is the one which will come in a direct contact with antigen. That is important point. Now you can ask me one more question. I will explain you in the upcoming slides. Sir, how the T helper cell come in contact with antigen with the help of macrophage? What the macrophage will do? Macrophage phagocytosize the antigen and it will express the antigen on which molecule? MHC molecule. To that MHC molecule, who will come and attach us? T helper cell will come and attach us. And I told you in the yesterday class, which MHC molecule T helper cell will come and associate friends. Every student remember MHC class 2. That is important for the exam. And T helper cell contain what kind of a surface marker? Very, very, very important. T helper cell is having a CD4 surface marker, not to get confused with this particular point. And friends, I told you very clearly to all the students, T helper cells are of two types. Listen friends, listen. One is called TH1 and another one is called TH2. I can tell you in a simple manner, TH1 will always associated with the macrophages. Why? Because antigen is associated with the macrophage only. So T helper cell associated with macrophage, TH1 activate the TH2. Then what the TH2 will do? TH2 will activate the B lymphocyte. Then B lymphocyte differentiated into plasma and memory cell. Then the plasma cell is going to produce the antibody. Simple concept, easy concept. Now you understand the two type of T helper cell. Now friends, every student try to understand me. Cytotoxic T cell, which we also call it as what friends? A TC cell. What is that? Cytotoxic cell or TC cell. This is more important for the exam. Why it is more important? Remember my words. So friends, this is the cytotoxic T cell. On the surface of the cytotoxic T cell, which surface marker is present? Every student remember, it contains the surface marker named as CD8. What does it contain? Very good, CD8. And CD8 is associated with what friends? That is also very important question for the exam. Suppose this is the macrophage. It is associated always with the MHC class 1. So this is important for the exam. Now let me ask you one very simple question to all of you. My question is, among these three cells, all the students has to understand me correctly. Among the T helper, among the cytotoxic T cell, among the T suppressor cells, which cell can inhibit virus infected cells? Listen my question very correctly. Which cells can destruct the virus infected cells? Wonderful. Very good, Srinivas. What is that? Cytotoxic T cell. No, Varaprasad, it is not. No, Renuka. See, I will tell you the difference here. Why you people got confused? Listen my question very carefully. If you understand the statement, it will be clear. In the exam, if they ask you a question, which of the following T cell recognize the virus infected cell? This is a cell which is infected by the virus. So which of the following cell is infected by the virus? So I mean to say, which is recognizing the virus infected cell? That is T helper. T helper is very good in recognition. But I have asked you a question, which T cell is involved in the killing the virus infected cell? That is the cytotoxic T cell. It's a very important question for the exam, not to get confused. Now friends, let me tell you one very interesting point to all of you. Suppose I told you, friends, when I complete the entire immunology, then only you will get a very good idea of immunology. Now, this is the cytotoxic T cell. This is a cell which is virus infected. Friends, remember my words. If the cytotoxic T cell want to kill this particular virus infected cell, all the students listen. First of all, cytotoxic T cell has to come and associated with the virus infected cells. if it is not recognizing or not associating the virus infected cell, it never ever destroy it. So friends, all the students listen my statements correctly. So this is what do we called cell mediated cytotoxicity. This is also they can ask the question in the exam. Which of the following T cell can show the cell interaction or cell mediated cytotoxicity? That is cytotoxic T cell only. If it want to destroy the targeted cell, it has to associated with the targeted cell compulsory. Now, I will tell you in the later concepts, but I'm just giving you one hint. How cytotoxic T cell will destroy the virus infected cell? By releasing lot of chemicals. What are those? By releasing interleukins, by releasing interferons, by releasing granzymes, by releasing lysozymes. So by releasing all those enzymes, cytotoxic T cell is going to destroy the virus infected cell. Never ever get confused with this particular point. So now all of you understand very clearly what is T helper cell, what is cytotoxic T cell. So friends, what is the last type of the T lymphocyte? T suppressor cell. Once again, friends, remember, let me ask you one simple question. Every student say yes, sir. Very good, Saidilu. Porphyrins, very good. Good. Now tell me, porphyrins also comes under the category of interleukins. Now my question to all the students is, I'm giving you one statement. T suppressor cells always active in the immune system. Tell me, Anupama, is this a true statement or a false statement? Sujata, every one of you. Venu, T suppressor cells are always active in the immune system. Undoubtedly, it is wrong statement. T suppressor cells are never ever active all the time, friends. It is most of the time. It is inactive. Not most of the time. It is always inactive. When we will activate the T suppressor cell, tell me. We will activate the T suppressor cell when we want to suppress the immune system. Let's take an example. Example. I am undergoing the kidney transplantation. Or I am undergoing the liver transplantation. That time, friends, suppose every student listen my words very correctly. I am undergoing the kidney transplantation. I have taken the, very good, I have taken kidney from my own brother. So 90% of the kidney will match. But sometimes our immune system, you know what they will do? Listen, everyone. Our immune system, which contain MHC molecule, we will discuss in the next slide. Very elaboratively, I'll explain you about the MHCs. Major histocompatibility proteins. What they will do, they will start rejecting the healthy donor tissue also. That time what happens? We have to stop this MHC. That time what we do? We will give some of the immunosuppressant drugs. What are the best widely given immunosuppressant drug? Every student commonly know that cyclosporin A. What we are going to give, what we are giving? We are giving cyclosporin A injection. Immediately cyclosporin A injection, what it will do? It activates the T suppressor cells in the body. Friends, the moment T suppressor cells get activated, immediately what is happening? It is going to suppress the entire immune system. By that, there will be a no graft rejection occur. The transplantation occurs successfully. But let me tell you one interesting point. What is that? Suppose, friends, kidney transplantation occurs successfully. You know what happened? After 3 months, in some cases, after 3 months, what happened? Once again, our MHC molecule start recognizing the donor kidney as a foreign substance. That time, once again, what happened? That time it start rejecting this kidney. Then once again, the kidney will become non-functional. That is the reason doctors will suggest to take the immunosuppressants drugs at least for the period of 6 months, on not on daily basis, weekly once or weekly twice, at a very lower doses. By that, in any case, if MHC molecules are recognizing the kidney as a foreign tissue, then immediately they will suppress it. Not to get confused with that point. Okay. So friends, listen everyone, look into this. So most of the students will study only B lymphocyte and T lymphocyte. They will leave the natural killer cell. Natural killer cell is also a very important cell. All the students listen to me correctly. From where the natural killer cell is derived, friends? Of course, natural killer cell is also derived from the lymphoid progenitor only. So friends, what's the most interesting point every student has to remember? Listen everyone. Friends, B lymphocyte compulsory required MHC. T lymphocyte compulsory required MHC. So the question which is asked in the exam many number of times, which of the following lymphocyte do not require MHC? This is the question asked in the exam, which is the natural killer cell. I will explain to all of you how exactly the natural killer cell will act actually. All the students remember my words. It's a very easy mechanism. Every one of you listen to me correctly. See friends, all the students listen. Suppose this is a cell which is infected by the virus. Remember everyone. If any natural killer cell wants to work in our body, it is a compulsory require immunoglobulin or antibody. Which antibody compulsory required for the natural killer cell? That is the immunoglobulin G. IgG is a very important antibody required for the natural killer cell functioning. Suppose this is the virus infected cell. Okay, you got it. Now the virus infected cell, who will recognize this? This is recognized by the immunoglobulin G. See friends, now every student listen. This is antibody, right? On the tail portion of the antibody, which portion? On the tail? On the tail portion of antibody, there is a receptor is present. What do we call that receptor? FC receptor. I will tell you in the later classes, fractional crystallizable receptor. So there is a presence of FC receptor. Now friends, this is what cell? Natural killer cell. On the surface of the natural killer cell, there is another receptor or the surface marker is present. What is that surface marker named as CD16? This is also important for the exam. So what I am trying to explain to all of you, the CD16 surface marker, which is present on the natural killer cell membrane, will come and associated with the FC receptor present on the IgG. Friends, let me tell you one very important point. Suppose example, IgG is not associated with the virus infected cell. Friends, then the FC receptor never attaches to the FC receptor. My point is very clear to all the students. Natural killer cell associated with the FC receptor of IgG only when the IgG recognize the virus infected cell only. So friends, what is my conclusion to all of you? Then a natural killer cell stimulate the IgG to destroy this virus infected cell very efficiently. So now what is my important statement here? If the natural killer cell want to show the function, what is required compulsory? What is required compulsory? Antibody required compulsory. So they can ask the question in the exam, which of the following immune cells can show the mechanism of ADCC? What is the mechanism of ADCC means? Antibody dependent cell mediated cytotoxicity. Yes or no? So natural killer cell is destroying the virus infected cell. There is no doubt about it. But how it is destroying the virus infected cell? With the help of antibody. That's why it is called ADCC. Once again, I'm reminding to all the students in the exam, when they ask you which of the following immune cell will show the cell to cell interaction, that is a cytotoxic T cell. And in the question, if they ask you which of the following is involved in the ADCC, that is what friends? Natural killer cells. Not to get confused with that point, easy to remember and easy to understand. So friends, what we completed so far? We just completed the first class, that is the common lymphoid progenitor. Now we are going to study second class, myeloid progenitor. See friends, you people no need to read very depth of the neutrophil, very depth of the new eosinophil, very depth of the basophil. Where we will study very depth. We will study under human physiology, under the blood circulation chapter. We will study very elaboratively. Now, what you people has to remember about these three, you have to remember their immunological functions. That is what we people has to focus. Now listen to me very correctly without confusion, all the students. So now we are going to study first phagocytes. What are the example of phagocyte? I told you phagocytic cells, monocyte and macrophage. Most of the students will get a confusion between, first of all, what is the process of phagocytosis? This is important question. What is called phagocytosis? All the students listen. Phagocytosis means engulfication of bacteria or engulfication of pathogen. Very good. No, Radhika, it's not like a solid food or a liquid food. That we will read in the cell biology. Solid food, that is different. That is the pinocytosis. Okay. That's little different. So now, whether it's the solid or liquid doesn't matter for us. If the pathogen can be destructed by the phagocytic cells, that is called as phagocytosis. You got it correctly. Now all the students listen for the exam. What is the difference between the monocyte and macrophage? I will explain you the mechanism also briefly. Focus. Both are called phagocytes only. Friends, all the students listen. Both are called phagocytes only. Listen my words. But what is the difference? Monocyte is only present in the blood circulation. When the monocyte from the blood circulation, it entered into the tissue, you can't call it as the monocyte. Why, friends? Because monocyte is completely differentiated. It is completely differentiated into what, friends? It is completely differentiated into the macrophage. Now, remember my words. Macrophages are also of different types, friends. How? Very good, very good. I will explain, Saidulu. Listen. Macrophages are also different types. We have given different names based on the tissue that they enter. Remember my words correctly. Based on which tissue it is entering, macrophages are given different names. You only listen. If the macrophage entered into the gut, you have to remember all this. It's extremely important for the exam. If they enter into the gut, we can call it intestinal macrophages. If they enter, very good. If they enter into the lung, we can call it alveolar macrophages. If they enter into the connective tissue, we can call it this is important. Now remember, Saidilu, we can call it histiocyte. Name of the macrophage which is present in the connective tissue is called histiocyte. Name of the macrophage present in the liver, we can call it Kupffer cell, not to get confused. Name of the macrophage present in the kidney, very important, mesangial cells. Same macrophage present in the brain, we can call microglial. Most of the students will get a confusion between mesangial and microglial. Never ever get confused. Mesangial is in the kidney, microglial is in the brain. What about microglial in the brain? What is the name of the macrophage present in the bone? We can call it osteoclast. I don't know what you do, friends. You have to remember every single example without fail compulsory. Now I will explain you how exactly macrophage is going to show the mechanism. This is more important for our exam. Because friends, our entire immune system starts working from this particular point only. If you understand this, most of the concept will be much very clear to all of you. Someone joined newly. Sudhakar, I think you joined newly. No, please continue. Please be on time. Now all the students, listen to me. How it is actually showing the mechanism. Understand everyone. This is the macrophage. See friends, listen everyone. You all people given the answer that macrophage is doing the process of engulfication or the phagocytosis. All the students listen how it is happening. Remember every one of you very correctly. Look into the picture. This is macrophage. All the students listen. Macrophage having a finger-like projections. You can see right this like this. What do we call that finger-like projections? We can call it pseudopodia. All the students follow me. With the help of pseudopodia, it will trap the bacteria. You can see that. You can see that picture. It is completely trapping the bacteria. Friends, the moment it trap the bacteria, immediately what happen? You know what process will occur? Every student remember, receptor mediated endocytosis is happening. All the students has to understand me. What is happening? Receptor mediated endocytosis means, friends, this is only receptor. Along with the receptor, the bacteria will be taken inside. You can see that. You can see it. So the bacteria is packed inside. The bacteria is packed inside the vesicle. What is the name of that particular vesicle? We called, friends, we called it as a phagosome. Very good. Now, friends, all the students listen. Now the phagosome combining with what? You only look into the picture. Phagosome is combining with the lysosome. When the phagosome combined with the lysosome, what do we call it as? We called it as phagolysosome. Phagosome plus lysosome called as phagolysosome. Now you listen the important concept. And we all people very well know lysosome contain abundant amount of the hydrolytic enzymes. Is it or not? Now lysosome release all the hydrolytic enzymes. They will break down the pathogen. Friends, which is important for the exam. After breaking down the pathogen, a little part of the pathogen, it is introducing on the MHC molecule. MHC is a protein which is present on the surface of the macrophage. And let me ask you one simple definition, one simple difference. What is the difference between pathogen, antigen, and epitope? This is very important, friends. Most of the people will get super confused here. What is pathogen? What is antigen? What is epitope? See, friends, pathogen means what? Disease causing microorganism. Whether it is a bacteria, virus, a fungi, whatever it may be, whichever causing the disease, that is called pathogen. Now, question is very clear. How they will cause the pathogen? Sorry, how they will cause the disease? By releasing antigen. So you can ask me, sir, what is antigen? Antigen may be anything. You take an example, bacteria. Bacteria release a cell wall. You take an example, virus. Virus, in order to show the action, it will release its RNA. Bacteria also release the DNA. Friends, whichever released by the bacteria, listen everyone, listen everyone. Whichever released by the pathogen in order to show their action, that is called antigen. But friends, the point is that, suppose I will explain in the MHC, that is how immunology is, all are interconnected. Suppose, friends, this is the antigen and this is the antibody. Every student commonly know that. We will study elaboratively. Antibody having a binding site. What do we call that binding site? Paratope. So to the paratope, antigen has to come and attaches. But you see this, antigen is very large. So this much large antigen never attaches. Only a small part of the antigen. What do we call that small part of the antigen? Epitope. Immunologically active part of the antigen. What is the definition I am giving you, friends? Immunologically active part of the antigen. Biologically active part of the antigen. We can call it epitope. That epitope will come and associated with the paratope of antibody, then antibody will destroy this. So always remember what is pathogen, what is antigen, and what is epitope. Anyway, all the students listen now. Now finally, friends, a little part of the antigen, where it is expressed on the MHC. Now you only tell me, which cell will come now? Undoubtedly, T helper cell will come. Yes or no? Now, T helper cell, this is class 2 MHC. Then, with the help of CD4 surface marker, the T helper cell will take the antigen. Friends, immediately, T helper cell get activated. Then, T helper cell release the interleukins. That interleukins activate which cell? B cell. Then, B cell differentiated into plasma cell, memory cell. Then plasma cell produce antibody. That antibody will come and destroy this particular antigen. This is what happens in our body whenever a pathogen enters inside. So The moment pathogen enter inside, macrophage become active. Macrophage trap the pathogen in the phagosome, combine with the lysosome. It will form the phagolysosome and it will break down the pathogen. Little part of the pathogen called antigen that is expressed on the MHC. Then there is activation of T helper cell, then activation of B cell, finally differentiation of plasma cell into antibodies. Then the antibody will come and destroy. This is what happens in our body. So that's the reason what I'm trying to explain you. In the immune system, the first important step is phagocytosis. When the phagocytosis occur, then only we can destroy the pathogen very easily, not to get confused with that particular point. Now, so friends, what we studied now, we understand what is monocyte, we understand what is macrophage. See friends, monocyte and macrophage is going to show same function. There will be no change in the 1% also. Function is same. But monocyte is always present in the blood. Macrophage is always present in the tissues. Next one. See friends, B lymphocyte, T lymphocyte, natural killer, these are all what kind of cells? They are agranular. The cytoplasm do not contain any kind of a granule. Now you look into the next one, granulocytes. Name itself indicating, right? When you observed under the light microscope, what you can observe, the cytoplasm contain granules. That's why we can call conspicuous granules. That's why we can call granulocyte. Neutrophil, eosinophil, basophil. I'll explain you briefly. Just listen. Friends, here we are not discussing about much about their structure. We are always discussing about with respect to our immune function. I will give you major lines to all the students. Remember, these are all very crucial because we can expect in the matching question. And I am requesting all of you to remember the number also. But number we will read in the blood circulation of the human physiology. Now let's talk immunology here. Remember the first point. Neutrophils are the first cells to enter at the site of inflammation. Suppose friends, remember everyone, this is my skin. My skin got cut. Then immediately all the blood cells will come there to initiate the process of inflammation. Among all, which will come first? Neutrophil will come first. So they are the first arrive at inflammation site. Always remember, first responders, early responders to the inflammation is neutrophil. And eosinophils. Eosinophils, friends, plays a key role in the parasitic infections. Like worm infections, eosinophils will place a major role. I'll explain you. Basophils plays a crucial role. Allergic and hypersensitive reactions. I am explaining about the structure also, but remember elaboratively about the functions with respect to the immunology. I will give you the differences also. Follow me. Friends, this is how the neutrophil look like. Now you only tell me, friends. The moment you look at the neutrophil, what you can observe, neutrophil is a multi-lobed nucleus. Yes or no? Of course, yes. Because when more than two, we always call it as multi-lobed. You only look into the picture. One lobe, two lobe, three lobe. So more than two, that's why we can call it multi-lobe. Not to get confused with that point. And friends, the cytoplasm of this neutrophil contain a lot of granules. You can see, friends, these are all granules only. Now you can ask me a question, sir, what does the granules consist of? All the students listen. Granules consist of enzymes. Friends, please don't get confused between lysosomes and lysozyme. Lysozyme is a single enzyme, only one enzyme. Lysosome is a big organelle. The granules contain lysozyme enzyme, peroxidase enzyme, collagenase enzyme. Remember, and what is the most important function, friends? All the students has to remember this. They are the one which first to arrive at a site of inflammation. Never ever get confused with this particular point. They will only come to the site of inflammation first. Friends, you remember natural killer cell is showing what mechanism? ADCC, antibody dependent cell mediated cytotoxicity. Friends, even neutrophils also can show the mechanism of ADCC. This is very important. You ought to remember. They can ask the question, which of the following cells will show the ADCC? I will tell you there are other cells also. So natural killer cells will show the ADCC. All the students remember, friends, remember my words. When the neutrophil is in the young condition, every student remember, when the neutrophil is in the young condition, there is only one or two lobes are present. That's all. When it is in the young condition, one or two lobes are present. So young neutrophil is called as band. Anything is same, friends. Young neutrophil is called as what? Band, because it contains only one or two lobules. But friends, when the neutrophil become matured, it contain many lobes. So when there is a many lobes are present, we can call it as a polymorphonuclear leukocyte. Sometimes, friends, you can also call polyp. Anything is same. You can call polyp, you can call polymorphonuclear leukocyte only when there are more than two lobes are there. Up to two lobes, you can call young neutrophil. What do you call that young neutrophil? You called it as the band. Not to get confused with that point, easy to remember. So now, next one, eosinophil. Friends, always try to compare one with other. It becomes very easy for us. You only look into the picture. If you see the neutrophil, one, two, three lobes are present. That's why I can multi-lobed. You look into the eosinophil, only one lobe, two lobe. Let me ask you one question to all of you. You called it as a bilobed. This is the one lobe, this is the second lobe. My question to all of you is, how the two lobes are connected with each other? Every student remember my words. Whether it is a two lobe, three lobe, four lobe, five lobe or six lobe, one lobe can be connected with the other lobe with the chromatin thread. Every student has to remember this. With the help of a chromatin thread, one lobe can be connected with the another lobe, not to get confused with that particular point. And friends, where the eosinophils can be distributed more on the skin, mucosa of bronchi, GIT. What did I told to all of you? Very good. Very good, Sudhakar. So they plays a crucial role where in the parasitic infection, worm infection, all worm infection, eosinophils will respond very fast. Friends, most of the times the questions will come from basophils. Most of the times. Now, let me tell you. Now, you only remember, neutrophil is what lobed, friends? Multi-lobed. Correct, na? And eosinophil is what lobed, friends? Bilobed. And what about the basophil, friends? Single lobed. Correct, na? And another very important statement for the exam, the granule size, this is the granule, right? The granule size of the eosinophil is larger than the neutrophil. The granule size of basophil is larger than the neutrophil and basophil. Sorry, neutrophil and eosinophil. So remember like this. Baso, eosino, neutro. Basophil having larger granule size, followed by eosinophil, followed by the neutrophils. Not to get confused, friends. It's very important and interesting one. The moment you heard the name of a basophil, remember my words, granule size is very large and it secretes many chemicals, not one or two. You only listen. It is going to secrete heparin. Every student commonly know that. What is a heparin, friends? Heparin is the natural anticoagulant. Then the next one is, what do we call, friends? Histamine. Every student know very well. We're going to study it. Histamine plays a key role in the allergic response. I will explain you how. The next one, it is also synthesizing PAF, platelet activating factor. What is the function of a platelet activating factor? Which is involved in the involved in the blood clotting, which is required for the platelet activation. So these are all synthesized from this particular basophil only. So why I called basophil is involved in the allergic reaction? Because there is a presence of the histamine. All the students remember my words. Not only basophils, even mast cells also going to secrete what? Going to secrete the histamine. Remember everyone, I already explained it to all of you. Friends, we really don't know from where the mast cell has come from. That's the reason I have written a statement that it is generated from unknown precursor. What, friends? It is generated from unknown precursor. So friends, every student listen, this is also very important for the exam. What is very important for the exam? What are the chemicals that are present in the mast cells? Every student remember, it contains histamine, it contains cytokines, it contains leukotrienes, it contains prostaglandins. Leukotrienes, prostaglandin, I will explain you in the inflammation process elaboratively to all of you. Friends, you all people has to understand me very correctly. Listen everyone what I'm trying to explain you. See friends, listen everyone. This is the granule. Inside the granule, all chemicals are present. Leukotrienes, prostaglandins, histamines are present. Now my point is, I want to break this granule. How can I break this granule? Whenever the antigen will come and activate the mast cell, then all the granules will break, all the chemicals will release. But the question is very clear. How this antigen associated with the mast cell? Every student remember, on the surface of the mast cell, which receptor is present? FC receptor is present. To the FC receptor, which antibody will come and attach us? This is also many times asked question in the exam, which is immunoglobulin E. So you have to understand my statement correctly. Don't think that the antigen or the pathogen will directly come and associated with the mast cell. That is wrong statement. Pathogen will come and associated with the IgE antibody present on the FC receptor of mast cell. Then IgE will get activated. Then what IgE will do? IgE will give the signal to all these granules to break. Once all these granules break, all the chemicals will be released. Then it will initiate the inflammation. You know, right, friends? What is the major function of a histamine? Every student remember my words. Histamine will place a crucial role in the constriction of bronchi. Every one of you listen my example very correctly. What I am saying, listen to me. Suppose when we go to a green park, when we go to a very fresh green park, some people all of sudden, they will get a continuous sneezing. Their eyes become completely reddish. Their eyes become completely watery. Why, friends? And they will get a very difficulty in breathing. All of a sudden. Why? Listen, every one of you. In the green park, there are many pollen grains are present. So all the pollen grains are freely moving in the air. Now, when we take that pollen grain, what happened? That pollen grains are acting as allergens. Now that allergens will go and activate the mast cell. Then mast cell release the histamine. How actually our bronchi look like? This is how the bronchi look like. So air is flowing like this. But now you see here, when the histamine got released, friends, listen. Now see the bronchi. Bronchi got completely constricted. Do you think air will flow? Never, ever. Then we feel extreme difficulty in the breathing. So when we come out of that particular place, once again the bronchi will come back to the normal position. Sometimes it won't reduce immediately. Every student know, right? Then what drug we will take, what medicine we will take? You will take on daily life. Antihistamine. What is the best example of antihistamine? Levocetirizine. Every one of you taken this medicine, right? A small medicine of 5 mg, levocetirizine. It act like a antihistamine. When we are having a continuous sneezing, obviously we will take this. It will suppress the level of histamine. Then sneezing also will be controlled. Anyway, remember that histamine plays a crucial role in blocking the or constricting the bronchi. The last and final one, dendritic cell. Okay, see friends, you remember I told you that macrophage involved in the process of phagocytosis. Similarly, friends, dendritic cells are also involved in the process of phagocytosis. No doubt about it. So now, why we called dendritic cell? The function is what? Don't get confused, friends. The function is phagocytosis only. But we are calling it as a dendritic cell On the membrane, it is having some extensions. You can see, right? The membrane extensions look like a dendrites of the neuron. Since they are looking like a dendrites of the neuron, that's why we are calling it as the dendritic cell. Friends, please not to get confused. Whatever the function performed by the macrophage, same function performed by the dendritic cell. There is no change at all. And you remember, I told you that there are different macrophages are given based on the tissue they enter. Same, based on the different tissue, we can call different dendritic cell. On the whatever the dendritic cells present in the skin, Langerhans dendritic cells. Whichever present on the lymph nodes, interdigitating. Whichever present on the monocyte derived dendritic cell. Whichever present on the lymphoid, plasmacytoid derived. I don't encourage any one of you to remember this. It's not required for JL or DL. Even it is not required. What is required most importantly, this one. That is type of macrophages that are present in the different tissues in the body. That is very important. So friends, what we studied so far, we just studied only small part. That is cells of the immune system. That is common lymphoid progenitor, myeloid, erythroid, megakaryocyte, erythroblast. So from tomorrow onwards, the real immunology will start. So where we are going to study all the barrier mechanisms like anatomical barrier, physical barrier, phagocytic barrier, inflammatory barrier. After this, we will study what is antigen, what is antibody, what is MHC molecule, everything we will study elaboratively. I will teach every single point of your syllabus briefly and elaboratively, but we will go little slowly the immunology. Other chapters we can go fast. See, human physiology I'll teach super fast. Evolution also I'll teach very fast, but we will do this very slowly because it is very conceptual one. Okay, clear, right, every one of you? Is that clear, everyone, without confusion? You can ask me, any one of you having any doubt. Anupama, is it clear for you? Sujata, clear? Okay. And once again, I'm reminding you, please don't ask me once again from tomorrow. I have already shown you, right? So tomorrow 8 p.m. class, attend from the zoology batch. You can see it after 15 minutes. And remember everyone, Sujata, Sudhakar, Anupama, and someone is there. Yeah, please message me once just to say hi. By that, I will add your numbers immediately to the new batch. So that zoology batch. So 8 p.m. class, 8 p.m. class always attend through that particular batch only. Sudhakar, eosinophil granules are larger. Sorry, first is basophil, very high. Second is eosinophil. Third is the neutrophil. Okay. Okay, friends, that's all for now. I will see you in the tomorrow 5 p.m. class. And please read because zoology syllabus is a vast syllabus, very depth. Please don't waste time. You have 98, sorry, you have 100, not 100. We have 110 days because our exam is on September 22, right? So make sure that you read all the concepts very thoroughly and very elaboratively. Once the chapter get over, I will only solve the MCQs and I will provide you maximum number of MCQs. Keep practicing all the MCQs. Then only you can crack the exam. Okay, perfect. I'll see you in the tomorrow sharp 5 p.m. Attend from the botany batch only. Okay. Thank you. Thank you all. Thank you.